So today is the last day we talk about bacteriology. 2 chapters left, and we'll finish today. Okay. The first one we want to talk about is anaerobic bacteria. We learned this before. Anaerobic means that no oxygen, right? Yeah, they are inhibited by the presence of oxygen. So this is a big group of bacteria which cannot survive in the presence of oxygen. Think about our body. Okay. You think about your own body. Even though we are living in this atmosphere with 21% of oxygen, actually in our body, there are a lot of parts which contains a low level of oxygen. Can you imagine the location? Such as deep in our tissue, the oxygen level is limited, right? Armpit, so usually it's low level of oxygen. And so some of them can be pathogenic in human and can cause trouble once they infect our body. And also our digestive tract is low level of oxygen. And in this big group, we'll learn several important ones. The spore-forming Clostridium species. The 3 major pathogens we'll introduce here: Clostridium tetani, Clostridium botulinum, and also Clostridium perfringens. And there are also some anaerobes which cannot form spores, non-spore forming bacteria. Most of them are normal flora, especially the members in our digestive tract, in the intestine. And they don't cause disease most of the time, but possibly they can cause some endogenous opportunistic infection. Okay. Today we'll focus on these 3: tetany, botulinum, and perfringens. Okay, here are some common features of this genus, Clostridium genus. All of them are anaerobic, so they don't require the presence of oxygen. Anaerobic means that they also have the respiration chain, the electron transport chain, but the final electron acceptor is not oxygen molecule, but other molecules. And this is after gram stain, you can see that they are gram-positive bacteria, right? Gram-positive. And they are, well, relatively larger than E. coli you saw before in the lab. So they are bigger and also wider, so it looks like the fat bacteria. And all of them can form endospores. Here, in this cell, this structure is the endospore structure. They produce toxins. Here the toxin means exotoxin, because they are gram-positive, so they can only produce the exotoxins. The natural habit is in the environment is the soil or the intestinal tract of animals and humans. And based on the species, it can cause different type of disease, like botulinum, tetanus, gas gangrene, and other infection. Okay, the common features. Okay, the position of the spore can be different. That depends on the species. For some bacteria, the spore is placed terminally, exactly on the terminus. Think about this is a bacilli. If the spore is exactly at the terminus, we call it terminally. Some just in the middle, so that's we call it centrally. Some are subterminally. Subterminally means that it's close to the terminus, but not exactly. And the spores are usually wider than the diameter of the rod. So like this. If you see, this is the terminal spore, and you can see the terminal, the spore is wider than the bacteria itself. And most of them are motile because they produce the peritrichous flagella. But there are also some exceptions, such as perfringens, it does not produce flagella. They grow under anaerobic condition, and they grow well on the blood-enriched medium, on media containing fresh red blood cell or blood components. So which means that those pathogens have evolved together with animals and humans for a long time. So they require the nutrients in the blood. And many Clostridia can produce beta hemolysis on blood agar. We learned hemolysis before that. We know the beta hemolysis is the complete destruction of red blood cells. So there is a clear zone you can notice. While the alpha hemolysis is this one, there is incomplete lysis of red blood cells and also reduction of hemoglobin to form the green pigment. While gamma hemolysis, okay, there is no hemolysis actually. Okay, the first one we want to introduce is Clostridium tetani, a bacteria which produces the terminal endospore here. You can see this is the endospore with multiple layer of substances. Okay, and the diameter here is bigger than the cell itself. Okay, terminal endospore to form the drumstick-like structure. Does anyone in this classroom play drums? Yeah? You can bring a drum next time. Drumstick. Yeah. Okay. And it's obligate anaerobic. So presence of oxygen is toxic to this bacteria. This bacteria tetany is actually everywhere in the soil. You can easily purify the bacteria, isolate the bacteria from the soil. We did that in the lab before, that you collect some soil, okay, treat it with high temperature to kill other bacteria, and then you leave the endospore to germinate so that you can get pure culture, the tetany. It does not ferment sugars, and it also does not digest proteins. So the nutrient requirement is special. It does not utilize sugar and proteins directly. Okay. Tetany produces an exotoxin called tetanus specimen. Tetanus specimen. This is exotoxin. And before we understand how this toxin affects our health, we want to recall what we learned in physiology class, how our immune cell, the motor neuron, control our muscle. So this is what happened. This is a motor neuron, okay? Motor neuron is able to release the neuron transmitter called acetylcholine. Acetylcholine binds to its receptor on the muscle cell, which can cause the muscle cell to contract. So that's how the motor neuron to cause the contraction of muscles. But you don't want the muscle cell contract all the time, right? You want to relax it when needed. So to relax the muscle cell, you need another neuron, the inhibitory neuron, to release the inhibitory transmitter, which can block the release of acetylcholine so that we can freely control our muscle to contract or relax. So that's in a physiological condition what happened. But once a patient is infected with tetany, the tetany releases the toxin. The story will be different. The bacteria grow in our body. It's not invasive, but it releases toxin, the tetanus specimen. The tetanus specimen is also AB structure exotoxin. It binds to the motor neuron membrane, the presynaptic membrane. Then the toxin is internalized. Then after that, the toxin can travel to the cell body of the motor neuron. Then it diffuses into the inhibitory neuron. In the inhibitory neuron, the toxin, is actually a peptidase, which can degrade protein molecules, degrade peptides, so that this tetanus specimen can degrade the snare protein on the vesicle. It blocks the fusion of the membrane, so it blocks the release of neuron transmitter. So that's how the tetanus specimen works. It blocks the release of inhibitory neuron transmitter so that the motor neuron cannot be controlled. There is no control, no inhibition of the motor neuron. Then the acetylcholine can be released all the time. It causes frequent contraction of the muscle cell. So that's how the tetanus specimen function in our body. This is a video here I can show you, but just prepare. This video is 7 minutes long, so it's a very nice cartoon. Sorry.
Tetanus is an acute, often fatal disease caused by a toxin produced by the bacterium Clostridium tetani. The toxin attacks the central nervous system, causing convulsive spasms of the muscles. Symptoms include lockjaw, stiffness of the lower limbs, and contraction of the neck and spine into an arch. These spasms are extremely painful and can cause bone fractures, tendon ruptures, and dislocations. Death often occurs due to respiratory failure or cardiac arrest. In newborns, the infection can enter via the umbilical stump in unhygienic conditions. Tetanus can also develop as a result of a deep puncture wound, as caused by stepping on a nail. It cannot be spread from person to person. Here we see the nail has entered deep into the muscle layer of the foot. The nail has introduced tetanus spores into the wound. Under conditions of low oxygen, as found in a deep puncture wound, the tetanus spores can germinate to produce living tetanus bacteria. The spores also release a toxin called tetanospasmin, which enters the bloodstream. Bacterial numbers increase as they grow and divide. Some bacterial cells develop spores at one end. The bacteria lives, releasing these spores and more tetanospasmin toxin. Bacteria without spores can also lyse to release toxin. The toxin enters the bloodstream. Movement is enabled by skeletal muscles, and skeletal muscles are controlled by motor neurons. An electrical message is sent down the axon of a motor neuron and arrives at the terminal buds. Chemical neurotransmitters are released, which bind to the muscle fibers, causing them to contract. Shortly after, the neurotransmitters are released from the muscle fibers, allowing them to relax. Repetition of this process tells the muscle fibers to contract and relax as required. If you are infected with tetanus bacteria, the tetanospasmin toxin can reach motor neurons via the bloodstream. The toxin enters the motor neuron via the terminal buds. The toxin then travels up the axon towards the neuron cell body, which is found in the spinal cord. After 2 to 14 days, the toxin reaches the motor neuron cell body in the spinal cord. Other toxins travelling in the bloodstream also enter motor neurons and make their way to the spinal cord. We will now zoom into the spinal cord tissue. In the spinal cord, the motor neurons are controlled by inhibitory interneurons. These interneurons prevent the motor neurons from constantly firing. Tetanospasmin toxins arriving in the motor neuron cell body interfere with the action of the inhibitory interneurons. To see how the toxin exerts its effect, we will zoom in on a synapse between a motor neuron and an inhibitory interneuron. The interneurons release inhibitory neurotransmitters into the synaptic junction. These prevent the firing of motor neurons so that the muscle fibers they control do not constantly contract. Tetanospasmin toxin moves out of the motor neuron across the synapse into the inhibitory interneuron. The toxin prevents the release of inhibitory neurotransmitters from the interneuron. The interneuron can no longer control the motor neuron, so the motor neuron fires constantly. Here we see many motor neurons have lost their inhibitory control and are constantly firing. This leads to continual release of neurotransmitters from the motor neuron terminal buds, which cause the muscle fibers to contract repeatedly until spasm occurs. The end result is the severe muscle spasms typical of tetanus. Tetanus is rare in most developed nations with comprehensive vaccination programs. With adequate immunisation, tetanus is a completely preventable disease. However, in the developing world, where vaccination is uncommon, tetanus is one of the top 10 causes of death. Many organisations are working to increase access to vaccination in developing nations, including vaccination during pregnancy and access to hygienic birthing conditions. You can protect yourself against tetanus by maintaining adequate immunity into adulthood by having a booster shot at least every 10 years.
Alright, playing this video is kind of time consuming, takes 7 minutes, but it tells us the story clearly how this tetanospasmin work in our body, how it causes trouble. It's important to understand and also remember the mechanism of this toxin. And later we'll talk about another toxin, the botulinum toxin. It's also a neuron toxin, but it functions in a different way. Okay. And now we know that this bacterial tetany is not invasive, so it does not spread in our body. It does not invade into the cell. And the disease is almost entirely due to the production of toxin, including toxemia. The toxin can also get into the bloodstream, which is also mentioned in the video. And because this is the anaerobic bacteria, the germination of the spore requires a special condition. So that's condition with low level of oxygen. Here are some factors which can aid the germination and toxin production, like necrotic tissue. A lot of cells die through necrosis, and also presence of calcium salts, and also associated with other co-infection, pathogenic infection. Can anyone give me the reason why the co-infection with other bacteria can help the germination and growth of Clostridium tetani? Anyone? Anyone know the reason? Because this pathogenic infection, a co-infection, the bacteria might use up the oxygen in a small environment, in a niche, so that it will establish a low oxidation reduction environment, which facilitates the tetany germination. Okay, so this is the pathogenesis. And tetany incubation can vary a lot, from several days, 4 to 5 days, to many weeks. That depends on a lot of factors, such as the depth of the wound, the number of spore contaminated, and also the distance between the wound to the central nervous system. It's all influence the incubation time. And symptoms of tetany infection, including the tonic contraction like this, and this is a newborn, the neonatal tetanus due to infection not a long time after born. Okay, so that's the clinical findings. For diagnosis, tetany infection diagnosis is quite different from other bacteria. When we introduce other bacteria, we want to know like what specimen to collect, how to analyze the specimen, how to culture the bacteria. So that's a standard procedure. But for tetany, we usually can tell the diagnosis based on the clinical symptoms, and also ask the patients about their history of injury. Like several weeks ago, they got infected, they got some injury, some deep wound, and they show the typical symptoms of tetany. So this, we don't have to collect specimen to culture the bacteria. Okay. Prevention and treatment. We can use active immunization with toxoid. We mentioned that before. The DPT vaccine, which contains the T means tetany, so that's the toxoid. And we should take proper care of the wounds contaminated with soil. Just in case that you go hiking, you play outside, you have some wound which is contaminated with soil, you want completely clean the wound. Ask the doctor to cut the wound open, make a surgical debridement to clean everything out. That's very important because in the soil, it contains the tetany spore. And you can also use the antitoxin, the neutralizing antibody, to protect emergent protection once there is, in case that there is toxin release immediately after injury. And you can also utilize the antimicrobial medicine like penicillin to inhibit the further growth of bacteria in the wound. Yeah. Okay. Immunization. The active immunization against tetany, we use the tetanus toxoid. So that's DPT, we mentioned a lot. So the initial immunization of DPT, little kids receive 3 injections before 1 year old. So the first year, they receive 3 injections. And 1 year later, they would receive another injection of DPT. So that's the initial immunization. I guess that's what happened in China, the kids would receive this. But in some countries, the kids would receive another booster injection once they go to school. So when they go to school, they would receive a booster injection because the little kids play a lot in school and they might have a higher chance of injury, so they would have this protection. I guess also in some of you guys in your country, you would get a booster injection every 10 years, right? Yeah. So that will provide a long-term protection or lifelong. So every 10 years, you receive another vaccine so that you are protected. But this policy is not used in China. In China, the adults won't receive the booster injection. And if you work in as a certain career, a certain field, you want to protect yourself. Like if you work as a researcher in a biological lab, you handle animals a lot. You want to protect yourself with a vaccine. You can receive a vaccine against tetanus. And like the garbage collector, they would have a higher risk of exposure. They would also receive the vaccine. Okay, the second pathogen we want to talk about is Clostridium perfringens. This is a typo. Okay, sorry. I actually updated the file. Perfringens, the P here should be little p, not a capital P. All right. This is also a spore-forming bacteria. The spore is formed subterminally. Okay. And it has capsule structure, and here you can see the presence of capsule surrounding the cell, and also here. Okay. But perfringen is not motile because they don't produce flagella. If you culture perfringens on blood agar, it produces a double zone beta hemolysis. Double zone beta hemolysis. It's like this. Okay, this is blood agar. In the middle, this is the bacteria. Okay, it's the inner zone. You can see the clear clearance, and you can also see an outer zone. Okay, And the two zones are due to the production of different molecules, while the inner zone, the complete lysis, is due to the theta toxin, that's the hemolytic zone, while the outer hemolytic zone here is due to the alpha toxin produced. Two different toxins. Perfringens is able to ferment a variety of sugars, different type of sugars. It's able to ferment. When it ferments sugars, it's able to produce both acid and gas. Acid means low pH, extremely low pH. And gas means that it can form a lot of bubbles. And perfringens is also able to digest proteins. So based on this property, there is a special experiment design called stormy fermentation. They use milk medium, so basically milk, inoculate the bacteria into the milk, culture the bacteria. Because in the milk, we know that it contains a lot of sugar, the lactose. So perfringen can degrade lactose, produce a lot of acid and gas. The acid will work on the protein molecule in the milk, okay, to denature the protein to form the acid clot, okay? And the large amount of gas produced will form a lot of bubble in the tube. So that you can see after fermentation, there are a lot of protein clots and a lot of bubble pushing up the clots. So it's called the stormy fermentation. This is the method to differentiate perfringen bacteria. Okay. Perfringens is able to produce different type of toxins and enzymes. Let me show you here in detail. This is the beta hemolysis, showing the double zone beta hemolysis. The outer zone, okay, alpha toxin, the outer zone. The alpha toxin is actually an enzyme called lecithinase, which is able to catalyze or degrade or hydrolyze lecithin, okay, hydrolyze lecithin on the cell membrane. So this is an important molecule. Lecithin is an important molecule on the cell membrane. Once the cell membrane molecule is degraded, is hydrolyzed, it causes the hemolysis of the alpha zone. While the inner zone is due to the theta toxin. Okay, the theta toxin can cause hemolysis and necrotizing of the cell. But it's a different molecule. It's not the lecithin molecule, lecithinase molecule. The bacteria is also able to produce DNase. DNase is the enzyme which degrades DNA, right? So we mentioned before that our DNA, once released from the cell, is sticky, which can trap pathogens. While the bacteria can degrade DNA so that it will help the bacteria to escape. And collagenase, which digests collagen molecule, the protein collagen of the subcutaneous tissue and muscle. Based on this, okay, toxin, alpha toxin, theta toxin, DNase, and collagenase, we know that perfringen should be invasive, right? It can degrade the tissue and spread, so it's invasive. It also produces enterotoxin, which causes intense diarrhea. And that's why perfringen is able to cause food poisoning. clinical findings of porphyrin infection. The major problem, also the lethal problem, is due to the gas gangrene, a special disease due to the tissue infection with porphyrin through contaminated wound. Like this person, he had some traffic accident, and from the accident site, the person got infected, and the porphyrin caused infection underneath the skin, and you can see the large area of necrosis of the tissue, and also the bacteria produce large amount of gas, forming a bubble on the skin. So that's we call it gas gangrene. The disease might develop in a rapidly progressing necrosis and causing fever, hemolysis, and produce and release large amount of toxin to form the toxemia, and later it will induce the patient's shock and death. So this disease is lethal. It's a deep infection into the tissue. You know, I don't know whether it's popular in your country or not. Acupuncture. Anyone know acupuncture? Yeah. Acupuncture is traditional medicine method. You use a needle, very thin needle. Yeah. Yeah, to get into underneath skin. So, well, they claim that acupuncture can solve a lot of problems like pain, cancer, a lot. But I don't really believe that. You know, acupuncture might help you to relieve some pain. Okay, that's true. But I don't think acupuncture can help person to cure cancer. But you know, when people do the acupuncture, you actually form some wound on the skin. So it's possible that if the needle is not well sterilized, it will induce bacteria into the tissue. It can cause infection. So I won't recommend anyone to receive that. Right? Okay, that's the lethal disease, gas gangrene. The second type of disease is the food poisoning, because the porphyrin is able to produce enterotoxin, which can cause intense diarrhea, and also okay, that's due to the ingestion of large amount of bacteria, and the diarrhea might last only a couple of days, and it's not lethal, and you don't have to treat the diarrhea. You just give them some symptomatic support, such as providing enough water and electrolytes. For diagnosis, okay, we can collect material from the wounds or the pus or the tissue, the necrotic tissue. And there you would find some very big, large gram-positive rod. Okay, to culture the bacteria, the one we mentioned just now is a milk medium, the stormy fermentation. So that will give you useful information. You can also use the blood agar, and on there you would find the double zone beta hemolysis. And you can also use the egg yolk agar. This relies on nuclear reaction. So this is the egg yolk agar. So basically you add in egg yolk to the agar. When it's solidified, you culture the bacteria on the surface. Because the bacteria is able to produce the alpha toxin, which is a lecithinase. And in the egg yolk, it contains a large amount of lecithin, so that surrounding the bacterial growth, you will see a halo of clearance of lecithin. So use this Nagler reaction, you can detect the presence of lecithinase by the bacteria. To treat porphyrin infection, we can use, first we want immediately help the patient to clean the wound, extensive surgical debridement of the involved area. You want to remove everything like necrotic tissue and the pus, everything, the gas, the liquid, everything, remove it. And then you can apply the antimicrobial drugs, particularly penicillin, to inhibit the bacterial growth. You can also give the patient some antitoxin to provide immediate protection. And those patients with large area of infection, you can also give them higher level of oxygen. So that's the hyperbaric oxygen. In this chamber, it contains a higher level of oxygen. And the oxygen would diffuse into the tissue, which can inhibit the bacterial growth. So that's also useful. And as I mentioned, for the food poisoning, we just give them support, give them like only symptomatic care. So, because the food poisoning usually can recover in a couple of days, it's self-limited. The last pathogen of Clostridium, the botulinum. And again, botulinum is a large gram-positive rod. And you can see here, that's the actual picture of botulinum. It produces spore, and the spore is subterminal, okay? Subterminal spore. This bacteria is motile because it produces peritrichous flagella. And botulinum spores are highly resistant to heat. So if it contaminates our food, and it probably takes a long time, like boiling 20 minutes, over 20 minutes to kill the spores. Okay. Botulinum also produces the exotoxin, the neurotoxin. The botulinum toxin is one of the most toxic substances ever found. But this neurotoxin functions in a different way than a tetany toxin. Let me show you. This is the normal condition we introduced just now. The motor neuron release acetylcholine to induce the muscle cell contraction. But the botulinum, okay, the red one, can block the release of acetylcholine. So that if the acetylcholine cannot be released, it cannot give signal to the muscle cell to contract. That will lead to paralysis. So your muscle cannot move, cannot contract. So that's the major problem that botulinum toxin causes paralysis. If this paralysis is only related to your like arm movement, it's not a big problem. But if the paralysis is related to the muscle cell involved in respiration, involved in ventilation, that will be a serious problem. Okay. But on the other side, the botulinum toxin can be used to make you look better. Okay, make you look beautiful. You promised to sit in the front line. Yeah. The botulinum toxin to induce the relaxation of muscle. So this is what happened. When we get old, we'll lose the fat underneath the skin, and also the amount of collagen will be also reduced, so that you would feel that the skin is not as good as when you were young. And also the muscle groups, okay, in the skin, the muscle might contract all the time, so that you will see the wrinkle, the big wrinkle on the surface of the skin. Okay, to help remove the wrinkles, we can use the toxin to find a proper group, muscle group, inject a minute amount of toxin to release, to relax the muscle, so that when the muscle is relaxed, the wrinkle is gone. So we can use this toxin to make you look better. Well, we know it works, but right now there is another problem, because this injection requires very tiny amount. So a lot of times the patient is overdosed. When they receive the injection, they are overdosed. So because this toxin is very toxic, and a lot of patients need to go to the hospital because they suffer from respiration or other problems because too much toxin injected. I guess earlier this year, one of our affiliated hospital, number 5 affiliated hospital, they report that within a short time, they received a lot of patients who are overdosed by this toxin. And if without treatment, this can be lethal, because the patient might suffer from respiration. So it's a big problem. Okay, yeah, most cases of botulinum results from the ingestion of food in which the Clostridium botulinum has grown and produced toxin. So both the bacterium and the toxin can cause trouble, especially in those food which stored in a condition with low level of oxygen, such as the spiced, smoked, or vacuumed food. It might contaminate with this anaerobic bacteria, leaving a lot of toxin production. Well, another case is for the little baby, the infants, especially before they are when they are less than 1 year old. So, the little infants, their digestive tract is not well developed. It's not well developed. The stomach acid is not high enough, so that once they ingest some food with botulinum contamination, such as honey, the bacteria can grow in the intestine. That can cause serious problem. Honey is welcomed in a lot of countries, a lot of cultures, and because before people know how to make sugar, honey is a very important sweetener. But just remember that in future, when you are a parent, don't give your newborn baby honey water, because it does not contain a lot of nutrients, just sugar, and it might contain some botulinum toxin and endospore, which can be lethal to the little kid. And when you are a doctor, just educate your patient about that. Okay. the toxin blocks the release of acetylcholine and causes the paralysis, the flaccid paralysis. And if okay, the paralysis can influence a lot of muscles. Usually, it begins with viral problem, viral disturbances. Like that's because of the incoordination of eye muscle, or they have trouble with double vision, or they have trouble with swallow and speech. Okay, the botulinum toxin, it can cause death if it influences the respiratory paralysis. Okay, so that's the muscle required for respiration or ventilation is influenced, and the patient cannot breathe well, cannot get in enough oxygen. So that's lethal. Or if the heart muscle, the cardiac muscle is involved, it can cause cardiac arrest. While the gastrointestinal symptoms usually not a regular big problem, it's not a regularly prominent, and there is no fever. Okay. Has anyone heard of any story in your life that some family or some people got toxified by botulinum? No. Well, that's good. So, like some people in your life, you hear of that they got some they had some food which contained botulinum. No, that's good. Because in China, there are a lot of traditional food. They make the food in a traditional way. You know, that's based on their experience. In some of those foods, it might be contaminated with botulinum, and if the family ate that special food, they probably will have trouble. A couple of years ago on news, it was reported that in a family they had some fermented tofu. You know tofu? It's prepared tofu, but fermented tofu. In those fermented tofu, it's an anaerobic condition, but that food was contaminated by botulinum, and that family ate that tofu, and several people died in that family. Okay. Laboratory test. So we collect the specimen. It can be like you can detect the presence of toxin in your patient's blood, or you can check the intestinal contents or the leftover food. That's an important material you want to keep to bring it to the hospital so that it can help the doctor to identify whether it's botulinum or not. And if there is toxin produced, you can use the toxin to inject into mice, and to see whether you can find the lethal effect. And also, if you have the antitoxin, the neutralizing antibody, you can see whether the antitoxin can specifically neutralize the effect of toxin. So this is a specific test. Okay. Hopefully you will never see such cases when you are a doctor in future. Okay. To treat the patient, we can provide the patient with immediate protection with antitoxin. So that's the anti-sera from horses. And those antitoxin can neutralize the toxins type A, B, and E, okay, that can provide immediate protection. And if your patient is suffering from respiration, ventilation problem, if necessary, provide them with proper ventilation, such as giving them mechanical support to help them to get in enough oxygen and help them to get enough gas exchange. So that's usually is carried out in any hospital, the respiration machine. And for our everyday life, if we eat some suspicious food, such as canned food, like or the spiced food, it's possible that in those food, especially the meat, it contains the toxin and also the spore. So we want to cook those foods thoroughly. One way we want to inactivate the existence of toxin in it, or that if there are live bacteria or endospore in the food, we want to try to kill them as much as possible, like boil them for at least 20 minutes. It's a long time boiling before we eat them. Okay, so that's all for the anaerobic bacteria.